Hi, this is Amy Proal with the Polybio podcast, and my guest today is Dr. Liisa Selin. She's a professor of pathology at the University of Massachusetts Medical School. And in her work as a viral immunologist, she and her colleague, Dr. Anna Gill, recently received an NIH grant to study the role of viral infection and T cell exhaustion in the development of ME/CFS. Their work may also shed light on the long COVID disease process. And with that, Liisa, hi, thank you for joining me.
Hi, Amy. It's very nice to be here.
Great. Well, Liisa, tell me more about your work. You and Anna have been doing work for a while now on particularly T cell responses in patients with ME/CFS. So what have you been finding?
So we basically feel that ME/CFS is a dysregulated immune response to viral infection. And it can actually be any number of different viruses. And I think we've all heard that in the past, if you're familiar with ME/CFS. Some people can get it after Epstein-Barr virus, which happens to be a major virus of interest for me in my research. And it truly can generate a very dysregulated immune response in those individuals who get acute infectious mononucleosis. But you can get ME/CFS as we consider it really a post-viral syndrome. And we now think that COVID-19, the individuals getting long COVID or long hauler COVID, is a similar phenomenon of a post-viral syndrome of a dysregulated immune response to the virus. And you end up with this symptom complex that I think we're all very familiar with exhaustion, post-exertional malaise, severe oxidative stress, and brain fog. And what we feel is the main mechanism behind it is that you end up with something known as CD8 T cell exhaustion. And I'm noticing as I talk to individuals in the field of ME/CFS, the individuals think that CD8 T cell exhaustion just means that the CD8 T cells are not working right. And that's all, that they're not making the right cytokines. But that's actually not what a viral immunologist means by CD8 T cell exhaustion. CD8 T cell exhaustion is actually a condition almost, and it encompasses a whole bunch of immunological defects, much like acute infectious mononucleosis does. Mm-hmm. So to get CD8 T cell exhaustion, you have to have a persistent antigen that hasn't cleared. Here, we don't know in ME/CFS exactly what the antigen might be, whether it's the original virus that you had, like Epstein-Barr virus, or is it that you cleared the virus like flu, because some people get it after flu. And what has happened is you developed exhaustion during flu and you reactivated all the persistent viruses that we have. You exhausted your CD8. And the most common persistent viruses that we have are the herpes viruses. And that does include Epstein-Barr virus. It can include cytomegalovirus. It can include zoster, which causes chickenpox and shingles. It can include a virus called HHV6, which is an infection we all get as babies, and pretty well 100% of people have that virus in their body. And that many of these viruses reactivate during immunosuppression, like when you're following transplant. So in a sense, you are in this immunological state where you are somewhat immunosuppressed by this exhaustion. However, at the same time, the CD8 T cell is exhausted, and what's known to happen is they turn on these inhibitory receptors on the CD8 cells. And to be part of that syndrome, you have an increase in these, and those receptors further dampen your CD8 responses, and they become even more exhausted. But on top of that, at the same time, there is this ongoing total response to the virus this persistent virus, whether it's your reactivated virus or the original virus, where you continue your antigen presenting cells, your innate response keeps going, and you keep making interferon type 1. Now your immune system is trying very, very hard to shut this down, because this is obviously not good for you. And when it tries to shut it down, it starts making a lot of IL-10 and TGF-beta. These are inhibitory cytokines. So now you have this soup that is very, very, very bad for you, that your immune system is super overactivated, it is super trying to shut itself down, and it gets stuck in that state. And that is not, as you can imagine, very good for you. And that is what causes all the symptoms and the disease, because one of the things known to happen with that soup of cytokines is that your metabolism gets totally messed up, and you end up in severe oxidative stress. And it's the classic oxidative stress that has been described very well for ME/CFS. And in fact, your CD8 T cells also end up in oxidative stress. And Maureen Hansen had a beautiful paper on how CD8 T cells are in oxidative stress in JCI recently in ME/CFS patients. So besides the metabolic effects, all these cytokines being turned on and being produced can also have major effects on your neurological system, because your brain is full of receptors for these cytokines that are produced during infection. And that's one of the reasons why we feel so unwell when we're sick. So that is what we mean when we say CD8 T cell exhaustion. And when I first realized that this is what was going on in ME/CFS, it suddenly totally made sense to me. And it kind of surprised me when I first, it dawned on me, because first of all, I'm a specialist in CD8 T cell exhaustion. I've been studying it for a long time in the classic LCMV model. So it might have occurred to me sooner, but also because one of the two things that we knew right off the bat back in 1984 when the disease was named was the CD8 T cells weren't functional, they were low in number, and the NK cells weren't functional and they were low in number. And that right there was an immediate clue that we should have been thinking about CD8 T cell exhaustion, but yet for some reason, we didn't get there. And we didn't really get there until, for me, until I saw this data. And this just leads me to reveal a bit about myself. Mm-hmm. To let you know that I have had ME/CFS for the past 45 years, since 1974, before they even knew what the disease was, and they did not know what was wrong with me. And you can imagine that was a horrendous time. I was in my first year of medical school. So that's not something I personally like to remember. And I have had 3 severe episodes more recently, a 4th one of ME/CFS, but fortunately for me, I have at least recovered to being functional in between. Although, as you can imagine with ME/CFS, each episode you lose some very important aspect of your life. And with my 3rd episode, I lost the ability to do clinical practice, and I just do research since 1996. You have to constantly, with the disease, prioritize where you're going to put the energy that you have, because your energy is compromised because there has been no treatment. But it's been in the back of my mind all along to try to understand, and I strongly suspected that a virus was involved. In my case, I suspected enterovirus, which can also be a persistent virus, which many people don't realize. When I recovered from those episodes, I kept thinking about what could be going on, but I went on with my career. And it was until recently when Mady Hornig had what I consider a seminal paper come out, her cytokine paper, and she showed this highly dysregulated cytokine response in ME/CFS patients that differed between the acute and chronic. And when it was one of those aha moments, I just went, okay, yes, that makes total sense. That's how I feel. I need to pursue this as much as I can. So in the background of my EBV research, we started looking at it. And one of the things I had noticed as we studied my blood routinely for other stuff in the lab in our virus work was that I had this unusual thing that nobody else seemed to have, was that I had increased CD4 positive, CD8 positive cells. And this brings in another very complex part of immunology that we know very little about. The most T cells are either CD4 or CD8. The CD4s interact with MHC class 2, and the CD8s interact with class 1. And they have very different functions. However, there's this tiny little population. Well, so in the thymus, before they mature, they're double positive. But once they've matured, they become one or the other, except for this tiny little 1% that seems to be in the peripheral blood. That it's not, no one knows whether it's coming out of the thymus or whether a CD4 cell suddenly upregulates CD8 or vice versa in the periphery. However, there are sporadic papers in the literature for the past 25 years showing it's a very heterogeneous population. They have overlapping functions between CD4, CD8, Treg, and also that they go up following some viral infections, and they go up in some autoimmune diseases, including multiple sclerosis, which is another neuroinflammatory disease. And I personally feel that ME/CFS falls in the category of neuroinflammatory diseases. And it is turning out we are working on MS and EBV at the moment, and other people are as well. And we are finding the immune responses in MS to be also abnormal to Epstein-Barr virus. And it's not, there seems to be some exhaustion of the important responses, but it looks like MS might be more focused on Epstein-Barr virus than the other herpes viruses, and even on specific epitopes. It doesn't look as broad as what ME/CFS is. So there's something, they're in the spectrum of a certain kind of a disease, but and MS does have the same problem as ME/CFS in the sense that people can go 5-7 years before anyone diagnoses what's wrong with them, because the symptoms are so vague and transitory. The neurological symptoms come and go. And once again, they too do not have a clear biomarker. There are some, and if you have it, fine, but if you don't, they also are in the unfortunate circumstance that they really do not have a clear biomarker that they can track for improvement from disease, which is the same thing for ME/CFS. So even if you come up with a treatment, you have nothing you can track other than symptoms that come and go. So that's how we got involved with the CD4 positive, CD8 positive cells, and it turned out 20% of my CD8s, and I'm still the highest we've found, were double positive. And this is highly abnormal in your peripheral blood. And we went back and looked at lots of other people we've done, and nobody else seemed to do it, have this population. So then we started looking at more ME/CFS patients, and lo and behold, there were many more. And it is also a spectrum, so that there's a variation anywhere from 2% to 20%, depending on the individual. And we need to understand more about the heterogeneity of the CD4, CD8s, what types they have. I, for instance, have a tremendous fraction of what are called regulatory CD4 positive, CD8 positive cells, compared to many of the other ME/CFS people who are much sicker than I am. So we don't know if that may be a beneficial thing. So there's a lot we need to learn about. So those are the two aspects. And it was following that, I asked Anna, maybe we should look at my CD8 function. And I did not expect to see what I saw. We just did a simple PNA ionomycin stimulation. And when she brought it back to me, only 3% of my CD8 cells were making interferon gamma. And at my age, that should be 50%. I was making no TNF at all. And at my age, that should be 35 to 40%. Because your memory cells should be easily activated by PMA ionomycin to make these things. So then we also started looking more at the other patients and found that once again, there's a spectrum of level of exhaustion, and we're looking for the exhaustion markers. And we do find that ME/CFS patients do have an increased expression of CTLA4, which is one of those inhibitory markers.
Oh, Liisa, I mean, my heart just surged when you're talking about this, because these are topics that are so important in ME/CFS. And in other words, I'm a microbiologist. I also study ME/CFS, and it's just cool to see a team just take the viral infections that we know many of these people sustain and correlate that with the immune response in a very straightforward fashion, understanding how the viruses, the activity of the viruses, are likely to impact the immune response. In other words, herpes viruses, like you mentioned, it's known, other teams have also shown that they can downregulate, in simple terms, the T cell responses, right? So it just made a lot of sense for you guys to study this straightforward trend that I'm gonna agree with you. I don't know why it hasn't been studied more robustly before your team, and to do it so carefully and so well, and also based even on your own personal experience in terms of being able to sort of really understand how what you're studying can fit in with the ME/CFS disease process that you yourself are so familiar with. And I think one of the, you mentioned a couple trends that I think need to be better understood in ME/CFS, which is that even if the infection that may have initiated your case doesn't necessarily persist, isn't necessarily sticking around, that insult, that infection let's say in simple terms, sort of dysregulate or take down the immune system in a manner that can allow pathogens you already harbor to reactivate and themselves drive new sets of symptoms. And I think that that's important for ME/CFS, even beyond the cases that are initiated by infection, because sometimes people report disease onset after a difficult pregnancy or a stressful event. But those are also events in which the immune system, in simple terms, goes down somewhat and allows for reactivation of other persistent pathogens, right? Especially the herpes viruses. So I think that you've hit on a key trend there, just by understanding that we don't have to always focus just on the onset. We can think of how that onset impacted the existing, what I'm going to call virome, that the patient already had. So that's really interesting. And I guess with that, let me ask you this. In some cases of ME/CFS, when you talk to the patients or even just look at the medical records, patients will report multiple infections. They'll say, I got sick, I had an Epstein-Barr infection, then I had another fever, then I was traveling, I had another issue here. Especially when it comes to the way that those pathogens can impact the T cell signaling and the T cell exhaustion that you're looking at. Do you think that multiple infections can feed into a case, and that, let's say you have an Epstein-Barr infection, and that's, leading to T cell exhaustion in a manner that you're starting to document. Now, when you get another infection, do you think that that makes it more likely that you'll struggle with that pathogen, which may in turn also lead to exhaustion, and in a way that almost snowballs the disease process?
Yes, those are all excellent points. I totally agree with you. And in fact, you're bringing me into my other area of specialty, which is known as heterologous immunity. And my whole career has been based on this concept that your prior history of infections influences how you respond to each new infection that you have. And whether that is the fact that you have cross-reactive T cells that get activated that are not appropriate for that infection, so that you end up with what's known as, you should be having a Th1 response for your CD4s and you suddenly have a Th2 because of your memory cells, that type of thing. And we've shown in mouse models very well that this can lead to very severe immunopathology, lung pathology, fat pathology, and in fact, that it can also at the same time lead to a totally asymptomatic infection because you clear the virus very quickly with those cross-reactive cells. But then because you cleared it too quickly, you may not have the right memory response to it. Or you may not completely clear it if it's some kind of persistent virus infection. And in fact, our research is highly quoted now during the COVID pandemic, because one of the things that we've always talked about is that it is your history of infections that you could have a totally asymptomatic infection, you could have a severe infection, or you could die. And we've clearly shown this in mouse models. It depended on what type of past history you had and what kind of T cell cross-reactivity you had. So that work is being very strongly quoted, which also brings me back to this issue in ME/CFS that the whole long COVID issue has also brought out, is that people always say, well, I didn't have an infection when I got my ME/CFS. Well, as we've seen with long COVID, many people who have no symptoms but have been infected are getting long COVID. And I know in my case, I did not have any kind of, other than the very first one, which is very unusual. The other ones, I just had a very mild viral illness with a bit of a sore throat and diarrhea. Other people who weren't physicians might have not even noticed they had it and not even think about it. And then 2 weeks later, boom, I'm nearly dead with ME/CFS, and it just hits really hard. And once you've had it once, I think we all know. I mean, the second time it hit me, I went to the doctor and I said, I know what this is. But at that point in 1984, ironically, that was the year that I had my second episode. We now had a name for the disease. And unfortunately, I had a doctor who was very familiar with it and was seeing patients and gave me excellent advice that has stood me for my lifetime for my dealing with the disease. But other people are not as fortunate with it. But that means even if you're not aware of having had an infection, you might have had an infection. Or as you say, it could be pregnancy, but even in pregnancy, you're immunosuppressed enough that you might have had an enterovirus infection or something else and not be even aware of it. And there were times in the past, besides enteroviruses, I did wonder about coronaviruses. So coronaviruses clearly came out with the first two, the SARS and the MERS. You know, they became very clear they were generating heterologous immune responses, as we call them. And that immediately made me more suspicious that they could also potentially induce ME/CFS, but I'm not sure that anyone has ever looked at that.
It's so interesting because, you know, there actually is a decent body of literature from the 70s, the 80s, where teams were looking at coronavirus persistence. There's one study, one team that found coronavirus genetic material and antigen in the multiple sclerosis brain. And in fact, in the demyelinating plaques, I can send you this study, they found coronavirus genetic material and protein. And then there's some more recent studies. There's another team that looked at tissue samples from people after autopsy, and coronavirus 229E was in certain tissue samples. So I agree with you. Sometimes especially with the RNA viruses, we like conventional teams sometimes don't think they're capable of persistence, but it really just isn't studied as robustly as it could be. And
Well,
like you said, if you're asymptomatic, which, I think when you mentioned the enteroviruses and some of the coronaviruses, that's where really exactly I agree with you, becomes interesting, because those, like SARS-CoV-2, are cases in which you may not even realize that you have that infection, and and you may be an asymptomatic case that then could progress to disease. So that's, yeah, it's a coronavirus trend as well. Yeah.
and very, so the reason why enteroviruses and coronaviruses are very attractive to the concept of heterologous immunity and T cell cross reactivity is that there are so many strains of
Yes.
this
Yes.
that have different T cell epitopes for the different HLAs. So the chances of you having epitopes that are slightly similar, but having an inappropriate memory response that gets triggered, that somehow leads to dysregulation, either clearing the virus too quickly, but it still is not the right response and you end up sick. Most of the time, I do believe heterologous immunity protects us. So
Mhm.
most of the time it decreases the viral load, clears it, and you do okay. But for some reason, if you have the wrong T cell receptor, I think in your cross-reactive response, whether you clear the virus quickly, if you end up with the wrong memory response, it may not be that time that you get sick, but the next time you get exposed to a variant of that virus,
Got it.
because
And
you're
just
going to be.
so I know that I'm understanding this correctly, what you mean is you may get a certain strain of an enterovirus, and
Yeah.
then your immune system attempts to create a, T cell or antibody response to that. Now,
Right.
it may happen too quickly, or that might be abrogated in a manner where it sort of, in simple terms, wasn't completely finished enough. And so, or you may have persistence of the pathogen either, or something like that. But in this one sense, you had a sort of incomplete immune response. And then the next time you get that, a similar, it might be a different strain of the same pathogen or something like that, the immune system comes in with that antibody response, but it's not well targeted and it's not complete. And so you get sort of a dysregulated inflammatory pattern that can then provoke a lot of ongoing issues.
What? Yes. So what
Okay.
you don't clear the antigen quickly, and it just starts driving kind of what we see in CD8 T cell exhaustion.
Right. Okay.
A massive immune response that's uncontrollable,
Right.
which can lead to death, as it does. And I do think that
Yeah.
is going on in some of the COVID.
I know. And in that model, in that sense, not model, but a little bit of virus can go a long way then, right? I think that's one of the things when people think about viral infection in a condition like ME/CFS, they go, why doesn't the patient have blatant encephalitis? Why? And it's like, well, no, a little bit of pathogen and a little bit of the immune response not being in, jiving can lead to pretty,
Yeah.
amplified dysregulation, right?
Yes. Yes.
Okay.
Yes.
That's
You have to have the immune system is a very fine-tuned instrument. It really is for pathogen, and I'm amazed at how well it does work.
right.
You can get a good immune response, clear your pathogen, and then you have these Treg cells also come in, shut it all down, and then it's over. But if that becomes dysregulated because things don't happen like they're supposed to, it can just escalate out of control. Or what can happen, which is what happens, which we have a whole paper on, that viral persistence can actually save your life. So in the LCMV model, if you have a really high dose of virus, of this special clone 13 virus, it completely exhausts your immune response. You end up in that state I described, but the mouse doesn't die. It just is persistently infected and probably has ME/CFS, but we don't study ME/CFS in mice. And it's possibly a model for ME/CFS that I would like someone to explore. But if you develop, we've tried a sort of medium dose of virus in these mice, and it just has to be the right dose. They develop what's called partial exhaustion. and which is what ME/CFS people look like. They're not totally exhausted, they're partially exhausted. And their CD8 T cells are able to function, and the immune system keeps driving on, and they die.
Hmm.
Whereas
Wow.
if you have a low dose of virus, you clear the virus and you're fine.
Yeah.
And NK cells are actually involved in that model. We have a Nature paper on that. So if you deplete NK cells in that high dose model, they become medium dose and die. Medium dose animals are if you deplete NK cells, they survive, because it turns out the NK cells are actually killing activated CD4 cells. And then that stops the CD8 T cells from getting help. They don't get enough IL-2. And that's what happens actually in the high dose model. The NK cells kill off the CD4 cells, and then the CD8 cells are totally exhausted. And see, NK cells are somehow involved in ME/CFS. We still need to figure out how.
Mm-hmm.
You know, and in the mouse model for persistent LCMV, if you deplete the NK cells after they become persistent, you can actually cure the virus.
Okay.
I don't know what happens to the symptoms of the mice because we don't study ME/CFS. But the other interesting thing this brings up is the other major human infection that is a persistent virus, clonal exhaustion infection, and that's hepatitis C virus. And nobody talks about that. And hepatitis C virus infection, they've recently got a drug that can completely cure the virus. And I've been recently talking to my colleagues in that field, and they say up to 50% of the patients, although they clear the virus, they still have an ME/CFS-like syndrome. So nobody has called symptoms in hepatitis C virus ME/CFS because they have a hepatitis C virus. ME/CFS people are people who don't know who the virus is or what the pathogen is. So, but I don't think people are even aware that hepatitis C virus infection patients, the chronic ones, have a syndrome like ME/CFS, but nobody calls it that.
I was actually aware of that. Do you think that there's any chance that when they target, sorry, I'm coughing, better not have a enterovirus. I'm just kidding. I feel fine. But when they clear the HCV, the hepatitis C virus, that the body having had to deal with that infection for a long time meant that the patient still has other viral, EBV and this and CMV that were still more problematic. And it almost is like you have to keep working on that after you even address the hepatitis C. I don't know. It just made
yes. So because they are actually globally, it's not that you're just exhausted to that pathogen. This syndrome that I described, it does result in sort of global CD8 exhaustion, as you can imagine. So they probably are having problems with other herpes viruses and pathogens. But I also am very, very concerned. It's going to be challenging in ME/CFS, even if we do treat the herpes viruses and we figure out and how many and which ones in an ME/CFS patient. I'm concerned that the imbalance, the dysregulation of both the metabolism, the neurological, it has been so long-term that it is just getting rid of that pathogen, and your microbiome,
yes,
is permanently dysregulated. And that's another aspect of this. And I think really, and this is what makes ME/CFS so challenging, is that you have to have a treatment that will not only clear whatever the pathogens are, but it has to address oxidative stress and the neurologic, plus I think the microbiome at the same time. And that does bring us to treatment. And I can say that I am also, although I'm new to the field of ME/CFS, although I've had it myself a long time, but for the research, we do have a drug that a small company that I'm working with right now. This drug's called Inspiratol. And the unique thing about this drug is that it can address at least three of these components at the same time. It is 5 antioxidants taken by nebulizer into the lung, and the lung is better than intravenous. It is just a massive vascular bed, and it has a direct connection to the brain that's not clearly understood. So things that go into the lung can get through the blood-brain barrier much faster. So this combination of antioxidants, actually, these have antiviral, antibacterial, antifungal components.
Yes.
They are great at reversing oxidative stress, because that's what antioxidants do. And at the same time, they are, interestingly enough, have an ability to modulate NF-kappa B signaling, which is huge because, as you know, that would then attenuate the activation of your T cells and could potentially reverse your CD8 T cell exhaustion. Obviously, this does not influence the microbiome, and that would have to be somehow potentially addressed another way, but it is hitting all, at least 3 arms and 4 arms, because it can also get into the brain rapidly to help with whatever's oxidative stress that is going on in the brain. And we have been having amazing success. This drug was initially designed to treat acute COVID and has already helped, I think, over 20 patients with acute COVID that have been treated experimentally. It's still very, very early, but they all improved their oxygen saturation within 12 hours from 80 to going straight up. None of them needed hospitalization. But then I got interested in this group through my colleague Ray Welsh, who was working with this group as a viral immunologist, because the FDA asked for a viral immunologist collaborator. And we started, because of what I knew about the oxidative stress of ME/CFS, I suggest we try it on LongCovid and ME/CFS. And we've so far, in a small group of people, they've tried it on 7. 3 LongCovids have done very well on it, are improving gradually with this treatment. And we've tried it on a couple of ME/CFS people who are also improving, and we're increasing the number, but we can't at the moment offer it any further treatment to people because we're still waiting for FDA approval. And until we have FDA approval, we won't have enough funds to get the huge clinical trials we need to get this done. But with everything that we've seen, and in fact, I'm one of the ME/CFS donors who tried it because I had a bad reaction to the vaccine, and I was just stunned when I took this medication because I probably would have required hospitalization from my response to the vaccine, especially because of the brain fog, could not retrieve words, could not speak. I was sleeping like 18 hours. I would pass out every time I ate. My head was bobbing up and down. I couldn't walk, and my lymph nodes were massive in my whole body. And in an hour and a half after taking the drug, the neurological symptoms had cleared, which basically I could think clearly. My splitting headache was gone. My head wasn't bobbing. I was sleeping. My energy was improved. It was, if I hadn't taken the drug, I would not have believed that this drug could do this. So that really pushed us on, and we're still, yeah. And I had a similar experience with a LongCovid. We also treated a close friend of mine who I was trying very hard to help because she was very sick. with long COVID. Also, she was in that group that when she got the vaccine, she got worse instead of better. Some people apparently get better, but she was in the group who ended up like me, actually. And she also is improving dramatically. And it's a slow progress. It's been 6 months that I've been on it, and you just keep getting better and better and better. Well, and we're setting up with Dr. Nancy Clemis to do a clinical trial with this. And we're still looking for funds. And this is actually, it is a lot of the money for my Classy website, if you've seen it at UMass, is being used to try to do the immunological studies on these patients, because they're incredibly expensive, and obviously they're not covered in my NIH grant, so I need to find funds elsewhere. And the amazing thing too is that we are seeing, we are able to track the immune response with this drug, and our exhaustion has been reversing with the treatment.
Wow.
As well as some of the other cytokine changes that we have. Yeah.
In other words, you've actually found a potential drug candidate that could potentially address some of the issues, and in some early anecdotal cases seems to be showing promise for helping to mitigate or actually impact these, what you're finding in terms of the infections and the T cell exhaustion. And now you're trying to raise funding to be able to do a clinical trial on patients with that drug. And then you will, as part of the trial, you'll be able to measure some of the key immunological parameters sort of before and after they take the drug.
Yes. And that would be, if we had something we could track and show that someone's improving and is also helping diagnosis, yes, that would really change both ME/CFS and long COVID. And of course, my grant does not cover the long COVID research. Mm-hmm. So I'm having to cover all that with either my own personal or donations. And I have to say, to get the long COVID work going, before we did this, I actually have had to make $100,000 donation myself to my own lab to keep that work going, because I'm determined we need to help these people, because we have to intervene, I think, in the first 2 years, because these people particularly are responding well to this drug. I think the long-term, highly dysregulated ME/CFS are going to be more challenging. We've done a little bit of work with them. They are showing improvement, but it's not as dramatic as we are with the long COVID. And as I've just said, I think we're going to have to come in from many different angles with the ME/CFS, with antivirals. And even, I believe, chronic Lyme is part of the spectrum. So we may have to come in with Lyme antibiotics. We have not yet tried Inspiratol on chronic Lyme patients, although a few of the ME/CFS patients that we've tried all seem to have a combination of diagnoses of chronic Lyme and ME/CFS, and it's not really clear. But I think it's going to be a challenge to reverse something that has been, like in me, 45 years.
Right.
Although I have to say, I was shocked that as soon as I went on the high dose of the Inspiratol, I have autonomic instability like many ME/CFS patients, and my resting heart rate varies 15 points, anywhere from 62 to 75. Like, I don't think normal people look like this on their Fitbit graph. And I went on the high dose, and it started varying 2 to 3 beats a day, which is just amazing. I haven't been that way, I don't think, ever. And my HRV has recently started getting into a normal range. Your heart rate variability, if people are familiar with that measure, another indication of autonomic instability, has reached borderline normal range, 27 compared to 14, 15, where I was running. So these are effects beyond the responses to the severe episode of ME/CFS that I got from the vaccine. These are also changes to my chronic disease. So I'm very hopeful about this drug.
Wow. That's huge, and I just want to clarify with long COVID, do you think that I'm not saying this is every long COVID case, but do you think that what's happening with some long COVID patients is actually the pattern we've already been mentioning, in which the SARS-CoV-2 infection, especially SARS-CoV-2 knocks down interferon signaling, and then all of a sudden you get that same, interferons control, for example, keep herpes viruses in latency. So you get that same reactivation of sort of what was already there, but not necessarily active, not necessarily driving as much disease. Do you think that that's sort of the reason that core trend is happening in at least some of these long COVID patients?
I think so, because 2 of the 3 long COVID patients that we looked at who have CMV, they clearly looked like their CMV had been reactivated. Their CMV CD8 responses were massive. Wow. I mean, I'd never seen anything like this other than acute CMV. And these were long-term people in their 40s that had CMV a long time ago. So we had very clear evidence that, and other people are reporting this, that and we're also noticing some reactivation, increase in EBV responses. And by the way, this is a very, very tricky area that's, I think, been confounding the ME/CFS field. So I noticed that they're constantly looking to try to measure the level of EBV or measure the level of CMV or HH36. But I really think you're not going to see dramatic changes in these viral loads such that we can measure it, because your immune system is doing everything it can to try and compensate for the exhaustion state. And I think part of that compensation is these CD4, CD8 positive cells that I'm talking about, because those cells actually are capable of making IL-9 and seem to be making IL-9 in most of the patients. And IL-9 is anti-apoptotic, so it would prevent them from dying. These CD4, CD8 cells are predominantly activated memory phenotype cells. They're not generally naive. And they seem to be a little bit more functional in the ME/CFS patients than their CD8s. So I think this is a way they're trying, one of the ways, I think there's a multitude of ways the immune system is trying to keep these pathogens under control, and maybe they just succeed, but their T cell responses might be the best way to actually try to look and see, or antibody responses. And I'm pretty sure everybody is aware, at least everyone I talk to says, oh, my EBV antibody response is off the chart. Well, there's probably a reason for that. Yeah. And we should be paying attention to that. I guess because so few immunologists so far have been involved in the field seems to be fixated on trying to measure these viral loads as a diagnostic, but I think you need to pay attention to the immune responses.
Absolutely. And coming from the microbiology viralis side on my end, one of the issues is that the viruses tend to persist in tissue and not even in the blood. Yeah. So the genetic material is rarely there to measure. So trying to measure from the blood it's just not, it's like looking for something in the wrong place. That's one of the things that's, there's not even that many virologists in the ME/CFS field, and we need more virologists as well, because a basic virology understanding will tell you that you don't even look for EBV in the blood. You look for it in the tissues it infects, or in the central nervous system, or, in the nerve. With some of the herpes viruses, especially HH36, they're nerve viruses, right? So they're nerve associated. So that makes a huge amount of sense to me, where, from our perspective, that's actually a challenge, because we're working with many living patients. So it's hard to find the viruses sometimes when they're in tissue. But like what you're doing, which is inferring the immune response to their activity, in blood and fluids that are more easily collected is really how we're going to figure out a lot of the answers on the side of that. So that's huge. Well, that's, yeah.
It is challenging
It
because
is.
the HLAs, to get these tests, these are very important tests, but to get them into routine lab tests is going to be something that can be done easily, that's cost effective. The things that we do, are very challenging, and right now we can't cover all HLAs. So but if we had people working on this,
Right,
get together and find a way to come up with a profile of how do we decide. I mean, they obviously know how to decide. So the immunosuppressed patient, like a transplant patient, they're so immunosuppressed that you can measure CMV, but even they really can't measure EBV. And even HHV6 can be difficult to measure. And if you see those immune responses activated, and we'd have to design, and these are not the easiest things to do, but certainly you need to do the antibody levels, decide and do a study and determine, how antibody levels correlate with diagnosis or severity of disease, which antibodies, maybe CMV and EBV. I mean, I hear this rumor that, and there's a lot of rumors in the ME/CFS field, and then I go look for a paper and there is nothing, that if you have both EBV and CMV, you do much worse. And I could see why. I hear this rumor, but there's no data. Nobody's looked. So, but of course, we still need the diagnostic, and I guess, it has been difficult to get the medical profession to even recognize ME/CFS as a disease. So it's understandable that some of these studies are not done, but there's so much that can be done and is doable.
Right. And to clarify then, and I think this is fairly obvious, but I'm just going to say it, that immune cell exhaustion, that you can document the differences in T cell function, the different responses. It's not going to turn up on a regular blood test when someone goes to their general practitioner. This is extra work that you do in your lab where you can find this. And what you're going to need to do is increasingly work with more immunologists to figure out how to basically document this and then make that into a panel of novel tests that can be done in a doctor's office. But it's not like a patient can walk into their doctor now and which is of course why too many patients go to their doctor and they say you're fine. So yeah, it's.
Oh, by the way, when I felt like I was recently dying and went on Inspiratol, my blood work looked totally normal.
There you go. Yeah. Okay. Interesting. Wow. And I'm sorry you had that experience. I'm just very glad that you were able to take this Inspiratol, and
work has looked normal every time I've had ME/CFS.
It's so frustrating. It's so yeah.
So routine blood work does not help. Apparently with ME/CFS, it's very little help.
Exactly. It really isn't.
Unfortunately.
All right. Well, yeah. Well, Liisa, this has been truly interesting to talk to you. I am interested and inspired by your work. I mean, you've gone and done everything from looking at, the viral side, the immune side. You have a great grasp of how those two factors, the infections and the immune system, sort of interact with each other. You're looking at the big picture of this. You're thinking about how multiple infections could play into cases. You're looking at core patterns that may impact patients with LongCovid and ME/CFS, with even components from the tick-borne pathogens being part of the picture. It's really cool to see that big picture thinking. And then it's increasingly exciting that you have a therapeutic that you think may be able to address this. So I'm so glad that you came on here and better explained this to me. And I really look forward to sharing this with more people. And I will definitely share it with your fundraising site, because this seems like work, and especially the clinical trial, that are worth pursuing. So thank you very much.
Thank you.